be a scanner, a sensor. Okay. So its purpose was to be used as a sensor, a scanner that can scan people's skin, their faces, and basically a medical device that will collect data, record data, and give you the results like you see on the picture at the bottom. So obviously it's a fictitious device, it doesn't exist. But my fascination with the device is that now with the advancement that we have in analytical science, it's not impossible to have a portable sensor-based device that can diagnose diseases by analyzing a large set of data and also collecting it. So I'll briefly talk about myself and what I do before I crack on with the subject that I really want to speak to you about. Like Aryaditi introduced me, I was a biomedical scientist nearly a decade ago, a bit over that. I trained during my doctoral training as an analytical chemist, and I used analytical techniques to try and find markers of Down syndrome. So it's a disease that is associated with mental deceleration. And it's especially the lifespan is quite reduced. People do live with it and have a good life because of really good support. So my thesis was in using analytical approaches to try and identify if early in the pregnancy, moms can know if they're carrying a Down syndrome baby or not. And that can give them an opportunity to try and think about the lifestyle they might have to rearrange. And that got me really fascinated with using different analytical approaches. So after my PhD, I started using lots of high-resolution instruments which generated lots of data. And it was that point when I kind of started learning about statistics and maths. And that's what chemometrics is all about. I'll talk to you about this in a bit. But I started using this data to understand with use of statistics and maths to make sense of it. So you might have heard of chemometrics in terms of machine learning. You might have heard terms like artificial intelligence, face recognition. They all kind of they are all the basis of chemometrics, but this field uses chemicals and mathematics together. And in 2017, I met Perdita Barron, who wanted to work with someone who could smell diseases. And the idea was to try and identify what the smell is or what makes things smell. And that's when I started the project on Parkinson. Since 2020, I've also taken on a task to try and eradicate TB. So it's using the same principle and same science that we have been doing since 2017. But the idea is to try and use the same platform to diagnose TB in early stages. And hopefully somewhere down the line, I expect to use that for personalized medicine. So each of one of us has a different sort of medicine for different diseases or even the same disease. So to briefly tell you about my entire research in a nutshell, I deal with small molecules. So these are molecules that are things like amino acids, tiny sugars, and they're really small in masses. So these squiggly things you see spinning on the screen are not so big. They're really tiny in terms of the chemical world. What I do is I detect these in biofluids, serum, plasma, urine, cerebrospinal fluid, name the biofluid, and I've probably done metabolomics on it. And what I do using that is I use predictive algorithms. I use mathematics to try and develop biomarkers that are predictive of things happening. So not things that are present in your body when you have a disease, but hopefully find markers that will be present in your body that will indicate somewhere down the line you might have a disease. And right now, my idea is to transfer these biomarkers on smart wearables that everyone is now very much used to. So things like smart watches, smart rings, patches that you can put on your skin, because these could be monitored actively and people are very proactive about their health using these devices. So where does it lead us? It leads us to a really healthy living. It leads us to trying to take control of our lives before things go wrong. And that's where I see the use of metabolomics that I do in a larger picture. So that's basically my research in a nutshell. So if you wanted, I could even finish the talk here, but I think obviously we are here to know more about this. And the kind of topics I want to cover today are all related to my research areas. I've spoken about metabolomics just a moment ago. I'll explain what it is, studying small molecules. I'll talk about chemometrics very briefly because no one likes statistics, especially not in a lecture for 1 hour. I'll talk about Parkinson's disease and tuberculosis, where I've applied these things and where we've got really good results of trying to monitor these diseases. And I'll talk about Sebomics, which is quite a new and upcoming field, which is study of sebum, which is produced from skin. So you might associate sebum with acne or pimples you might have had in your teenage. And that's the overproduction of sebum that causes it. But it's not all bad, and not everyone has been looking at sebum apart from skin diseases. But in our recent work, especially Parkinson and tuberculosis, we have managed to show that sebum has really good diagnostic properties for diseases that are not related to even skin, or they don't even happen on your skin. And to do that, I use lots of analytical powerhouses, like I use chromatography. You might have done it in your schools. I use mass spectrometry, which are quite high resolution instruments, and they can detect carbons and nitrogens in a molecule, and you can count them. And I also sometimes use spectroscopy, which is shining light on things. So there's lots of options I use to try and detect these molecules. So now that you kind of briefly know about what I do, I want to talk about the terms I just used. So let's talk a bit about metabolomics and what it is. I gave a hint saying it's a study of small molecules, but how do you measure these small molecules and why are these important? So the biologists in the audience might know the central dogma of biology, which is that we inherit genes from our parents, and what we are and what we are made is pretty much kind of in that genetic information. So our parents passed this on, we have no choice. This is just what passed on. So for example, genetic diseases like diabetes, you don't have a choice. If they're in your genes, you're more likely to have it. This information is transcribed, and it's studied by something called transcriptome. And they're transcribed to make proteins, which these proteins are the ones that kind of guide and make diseases happen or stop. And how do these proteins come into existence? Using the information that's transcribed from the genes that we inherit. So that's the central dogma of biology, that information goes from one end to the other. However, one of the key elements that's not considered in this central dogma of biology is metabolites. So these are small molecules that happen or that become to happen because of the changes in proteins, because of changes in transcription factors. And this is the part which is right at the bottom of the pyramid. If you look at all these molecules in a pyramid, there are lots of them, and they interact with each other and they change. So if I was to show you a picture of what metabolites are like, these are all the pathways in which they appear. So this is everything we know about human metabolome, and we know about sugars, we know about lipids, we know about amino acids, we know about nucleotides, and they all interact with each other all the time, irrespective of you being healthy or you being suffering from a disease. And the reason this is quite important is because whatever changes happen in your metabolites is the closest to your phenotype. So as scientists, we talk about phenotype as something that's more than just the appearance. So when I say phenotype, it's not talking about your eyes or how long your nose is, or your skin complexion. Your phenotype is not just how it makes you look, but how your body also looks from inside. And this phenotype is closest to how things change in terms of metabolites, not just proteins or not just your genes. And that's why studying them is really important, because the smallest change in metabolites talks about the largest change that could have happened in your gene or your transcript. So how do we study them? Now in your schools, if you had taken chemistry, or if it was one of the classes where you're given a piece of paper and asked to put some inks on it and put it in a bit of liquid, which is a solvent, you would remember that they traveled on the piece of paper like magic. And that's paper chromatography. What we use to study these molecules is liquid or gas chromatography, where we make molecules travel through a column, which is like a steel rod. and they separate industrial road based on how much they like the column. So to make it very simple, today's India-Pakistan cricket match. So if you're really a big cricket fan, and if you're sitting in my living room, you'll probably be here at 3 o'clock my time, and probably 7 o'clock your time, 7:30, because you know the match is going to be, it's about to start. So your affinity to my sofa, which could be a stationary phase, is really high. And the likelihood of you being at my place would be really long. Whereas if you had nothing to do with cricket, you hated it, the likelihood of you leaving my house in next hour would be really high because you have other things to do. So your affinity to my stationary phase, which is my sofa, would be really low. And the same thing applies with chemicals here. So if they really like my stationary phase, they will bind to it and they wouldn't come out too early. So if I show it as an animation, think of red circles and triangles, blue triangles as two different kinds of chemicals. And you can see they separated each other because the red circles really had high affinity to the stationary phase, whereas the triangles didn't have the affinity, so they left the column or the stationary phase really quick. When they do this, they separate themselves out. So now try and put this in perspective of our blood sample. Obviously, our blood is not made of just two molecules, but let's imagine it was just made of these two molecules. So there was no way to separate them right at the start before I ran the animation. But now that they've separated on the column, you can see if the detector if it was anything, say the detector is just your eye, so you can see the triangle. So you can see the triangles coming out first on the right-hand side, and then you'll see the circles coming out. And we use the same thing with the chemicals because we need to separate them. If we don't, this is what they look like. So this is something I've done on gas chromatography, and this is from a single yeast cell. So each of the peak that you see there, it's more than one analyte in there. But say if I didn't separate them on a column using the same thing I just showed you, they would all be like a huge blob right there. You'll see massive blue, one single peak like a mountain. But now you see them separate. So now I can see the things that are present in a single yeast cell. And even though the peaks, if you sit and count them right now, there are about 35, 36 peaks. But these peaks relate to nearly 100 metabolites present in yeast. And this is a single yeast cell, which is a very small molecule. But when you start looking at human serum or human blood, these are the sort of chromatograms you get. So these are the kind of graphs you make using chromatography. And each of the peak is more than one molecule in there. And this makes you appreciate how complex human metabolome can be, because these are the metabolites that are present in a single microliter of our blood. And without separating them, there would be no way we could study them. So we use this approach to separate all the metabolites that are present in our blood or our biofluids. So to just give you an example of one of the acids, hippuric acid, it's present in all our bloods. And if you study them using mass spectrometry, what you can do is you can break it down. So you see the chemical structure there of one of these hippuric acid. By looking at the chemical structure, obviously, if you're not a chemist, none of it makes sense. But the way I want to put it is, when you do mass spectrometry, you can break these things down. So, the end of OH and H or NH, they all got some kind of mass because all the chemicals measure to some mass. And what you can do is you can measure these masses, like the graph you see underneath, 77, 105, they are the weights or mass of these molecules. And when you start breaking them down, you can count the number of carbons, you can count the number of hydrogens that are present within that molecule. So say if there was something that that's completely unknown to us, it's an unknown marker, we don't know about this molecule, but we will still be able to count how many carbons, hydrogens, nitrogens, or oxygen make up this unknown molecule. And once the chemists know how many carbons are there, it's easy for them to make a structure of this molecule that's unknown to us. And then we know if a biomarker, even if we don't know what the molecule is, we know the structure of this biomarker. And we do this by matching things to different libraries that people generate online. So there are about 8,000 known compounds that things get matched to, for example, on GCMS. So that's the simplest way to explain how things are measured in blood. And using that, we can study things like small molecules that are related to very primary human energy metabolism. So these are the things that change when your body or your cells need energy. It could be any energy. So if you are running, you lose your ATPs, and you need to make these energies. You break down your sugars, you break down your larger carbohydrates, and you get this energy. You use GCMS, so gas chromatography, to kind of measure these sort of molecules. And you can also use something called liquid chromatography to measure larger molecules like lipids, which I'll talk about in a bit, and things like the molecules that are made from drugs. So if you are taking lots of medication and if you have got some kind of toxicity, that could be measured using liquid chromatography. So I use both of them to understand what's going on in things like Parkinson and tuberculosis. So I've talked a lot about the analytical part of it, but despite being able to do all this, what you can see in this picture is the summary of what I call a rain cycle. When we use all these biomarkers together with things like smart watches, smart glasses, as you see in the picture, the man under the tree, and when we combine both of these, what we get is the knowledge of how things are changing on the surface of human body and how things are changing inside the human body. And the scrolling table you see on your screen is the list of biomarkers that are proposed using metabolomic studies in the last decade or so. So there are about 68 good studies that proposed about 240 biomarkers for nearly 17 diseases. Some of them are very common, like Alzheimer's, diabetes, lung cancer. But these markers have never made it to the clinical trials. They have never made it to the end users. And that begs the question, why is it? Is it because we are measuring the wrong biofluid? Is it because we're measuring the wrong kind of analytes? Is it because we're measuring wrong molecules? And to understand that, I use something called Chemometrics, which looks at patterns of things. So you probably can associate this with face recognition. So if I give you an example, let's say if you're looking at this bit of face, an eye, it would be hard for you or anyone to understand who this person could be. If I showed you a bit of hair, maybe you had some idea that it's a person with certain kind of hair. If I give you another feature, nose, maybe you can put three together and you have a guess who this person is. But it's often difficult when two people are very alike in how they look, how they behave. For example, in this case, Boris Johnson and Trump. And even a good human eye could probably get confused. And it's the same theory with chemicals and different molecules that we measure. It's often very easy to confuse between molecules that behave in a very similar way. They appear in very similar fashion in different diseases. And you can't make sense of them if you just look at one particular feature. For example, here it was just an eye or a bit of hair or nose. You need to have the full picture. You need to try and understand how and where these molecules are placed, so that you can compare it against the other person. And then you know that despite them behaving similarly in Parkinson and a normal human, they are very different. And this is the theory we use to understand the interaction of molecules. And that is where I get most of my joy. And Joy is a lady that I spoke about, I think, earlier. So Joy is the person who can smell Parkinson's. She approached us in one of the Parkinson's meeting, and she asked a very interesting question to us. And the question was, can you smell Parkinson's? So I won't tell much about her. I want you to look at one of the videos, and I hope it plays fine given the bandwidth. But if not, I'll send the link to our editor and you can circulate them to everyone else. But this is the story of Joy.
I'm in a tiny, tiny branch of the population, somewhere between a dog and a human. And I didn't think it was an insult. Oh, that's quite nice. I can smell better than most humans. I'm Joy Milne. I'm 67 and I smell Parkinson's. My husband was a doctor. I was a nurse. I sensed a strange smell when he was around, and caused a bit of contention because I said he wasn't brushing his teeth and he wasn't showering and so on and so forth. So I just tried to be quiet. But then we went to a Parkinson's meeting and my nose just thought, wow.
This experiment is to see whether Joy can distinguish the Parkinson smells from the samples that we've taken from patients as they're separated. And if she smells it, we'll then know right away what that molecule is.
It's heavy, thick musk smell, very different. People with Parkinson's and their carers and their families, they will tell you that smell is there. I smelled it 10 to 12 years before I was diagnosed. As the Parkinson's got worse, the smell got worse, and it became just part of him. But I, with my sensitive sense, I could smell it all the time. It became quite uncomfortable, really. But I had the sense not to nag too much. We just put, Les, would like to thank you for your friendship and many happy memories over the years. That's his funeral.
God, you're a wonder, Joy. I mean, that time when you smelt Les on other people, and then now we're here, it's sort of amazing.
But best we've done, it really is.
And the background.
It's horrible watching your partner change like that. It really is. If we get the test right, they will never get to that stage, because at that stage of diagnosis, 60 to 70% of damage, neural damage, is already done. You're not going to find a cure at the moment. It's not there. But to stop or alleviate it is just going to be tremendous.
Right, so that's the story of Joy. You probably would have come across Parkinson's, and you probably know about Parkinson's more than I do. But like Joy said, and like we all know, there is no cure to Parkinson's. And when Joy met us, her question was very simple, that what she was smelling, has that been looked at? And no, it wasn't looked at that time. And there was no clean idea about how can we look at it. And that's when Purdy and I decided that obviously the approach of metabolomics is one that we could use to try and investigate what the smell is. So what we did, what you see in this little animation on the screen is again gas chromatography that I really rapidly gave a short introduction to. And what we did was we took samples from people's back using a medical gauze, the thing you use to put when someone bleeds. We used that to sample someone's back. We asked them to rub it on the back and put it in a bag and send it to us. When they did that, what they were doing was they were collecting sebum that's produced on your skin surface. This really oily, lipid-rich, waxy biofluid, you can't see it, but you've sampled in it, and it can be, it can trap lots of molecules. So what we did then was we put it in a gas chromatography where we put a sample in literally a bottle, and we heated up the piece of cloth that was sent to us. And as you heat it, what happens is the molecules, like you see in the animation right now, they evaporate from that piece of gauze and they go into something called thermal desorption chamber, where things are held for a long time and separated, and they go into a gas chromatography column where, like I said, based on their affinity, some molecules will go through the detector first, some will stay on there for a long time, and they'll go in later. And as they go to the detector, what we saw on the screen were the peaks that you see in different colors in that animation. But what we did was, while things were going into our analytical detector, we split the flow so that one part of it was going to the E-nose. So what you see on the top right corner where Joy has got a white thing, that's the E-nose. And what E-nose does is it gives Joy the ability to smell these chemicals while they're being separated. So every time a yellow peak appears, like in this animation, Joy can tell us what it smells like, and she can also rate the intensity of it. And when the red one comes in, she can again describe it. So she would be there with her headphone and mic telling us it smells like biscuit, it smells like burnt toast, it smells like pus, it smells like rice, and she can describe what it is, and she can use a button to say how intense it is. So using that information, what we did was we published this paper, and this was the very first paper that looked at skin biomarkers from sebum for any disease. And it got good traction. Like you can see, it's been viewed nearly 50,000 times by now. And it's been well accepted because of the novel work that was put into it. And the novelty lies in being able to detect things that smell on skin, and not only just detect it, but also being able to separate it and use it as marker for a disease. So the curve you see there, it's an ROC curve. What it basically shows is if you get a straight line in both ways, and if it goes to 100, you can detect a disease with 100% confidence. Obviously, you can see it's not good. So it's about 78% confidence we have using these markers, but then it was the very first trial. So even in the very first trial, we were able to detect difference between Parkinson's skin and control skin just based on the smelly molecules with 78% confidence. So we followed this up and we did another study where we tried and validated it in different people. So we now took sample from different people, totally unrelated to this trial, and we tried to find the same markers again and see if they were present. And again, you see that curve that I mentioned earlier in the previous slide. And this time we could do it with about 88% ability. So now we knew what we were looking for. So we could specifically target and we can get rid of lots of noise. And we could improve our method to do the same thing. And what you see on the right side this time is the interesting column is where it says VIP. So if you were to use two smelly components, we get about 75% confidence in detecting Parkinson's against control. If you use 3 of them, that increases to 80. And in the similar fashion, if we use 12 of them, we could get 85% confidence. So what it told us was that the smell wasn't limited to one marker, or it wasn't one chemical that was causing the smell of Parkinson. The smell of Parkinson is probably a result of interaction of more than one molecule in such a way that the smell comes off more stronger than it would normally in a control sample. And that's when we decided, and that's when we thought that, what we are trying to understand here is more complex than, just measuring a rose kind of smell or a musk kind of smell, because it's not one molecule that will cause it. Some things will go down to let the smell be, sensed as a really strong smell. So it won't always be a molecule going really high. It is probably that some molecules are going down, a few are coming up, and that up and down is causing the smell to appear. So then what we did is we followed it up by looking at lipids that are produced on skin. So I say it's quite a lipid-rich media. It's got lots of lipids in it. So what we did was we tried and forget about smell for a bit. We decided to just look at lipids that are present on the skin. We sampled the people in the same way. We asked them to rub on the back, send it to us. And this time, instead of looking at smell, which Joy could smell, we started looking at lipids, which are large molecules. And based on that, what we found was that there is a dysregulation of how lipids are produced on the skin. And it doesn't just happen when Parkinson's starts or when there is an onset of the disease. It increases. So the top right corner where you see drug naive and medicated PD are two groups we looked at. We looked at people who had just been diagnosed with Parkinson and had no drugs given to them. So they are drug naive. And the non-drug naive, the medicated Parkinson people are the ones who have been living with Parkinson for nearly 5 years, 6 years, and they've been on some kind of medication to kind of mitigate for this effect. And the size of the circle that you see is the effect of the change in lipids or change in particular kind of molecules that happens over time. So drug naive, so if I went to a clinic today, based on my rigidity of my body, my movement, a clinician will say, you probably have Parkinson. So I'll be drug naive. And for 5 years, if I take drugs, my Parkinson is not going to stop. It's going to keep getting worse and worse and worse. But I'm on drugs to mitigate my rigidity, my body movement. So I feel still better, but internally my Parkinson's is getting worse. And that you can see by the size of the circle. For example, sphingolipid are kind of lipids that are present in our cell membranes. And you can see that we detect more and more of these. We detect something called Carnitine Shuttle as well. And you can see certain things stay the same. For example, the way we produce fatty acids, it's pretty much same if you're drug naive or 5 years after you have Parkinson. So this study gave us insight into how things are changing, not just in the terms of smell, but also in the basic composition of lipids and sebum that's being secreted from our skin. And at this point, you'll probably say, hold on, Parkinson is, a neurodegenerative disease. It happens in brain, and you're looking at something on your skin that they're totally unrelated. And it's a good question if you're thinking of it, because we thought of that too. And that's when we came up with a hypothesis that we are seeing something that's probably present in all of us, but it's changing only when Parkinson occurs. So the graph you see here is currently in terms of clinical Parkinson's between stage 3-4 and stage 5-6 is where we can detect Parkinson. The redness in the brain shows how much damage is already done. So, 60 to 70% damage that Joy spoke about, that's stage 5 or 6. And you can see there's pretty much 20 or 30% good brain cells left. What we want to do is we want to push that from stage 5 and 6 to stage 3 and 4, where you see the pink line, where it's just about to start. And you can detect this if you can understand what lipids are so different. Obviously, that's where the current diagnosis of what we are proposing can do. But what we really want to push it to is the pre-clinical diagnosis stage, where the damage is not even occurring yet, but the markers, because the lipids will change, can be detected on skin, will tell us what will go wrong in brain before it goes wrong. So on the right, the graphs, you can see the only interesting part I will try and highlight in this slide is the blue versus purple colored graphs. So you see the arrow, I'll try and get the laser out. You see these peaks here where my laser is. They are present in Parkinson. So they are the kind of lipids that are present in Parkinson. But you see on the purple side at the same place, we don't see any of those lipids at all. They're absent in controls. So using this study, we were able to develop a way to detect Parkinson by just using a piece of paper. So now moving on from those, what we have done is we have developed a test where you literally take any A4 size paper from a notebook, take your sebum, send to us, and in 3 minutes, we can detect if these lipids are present against the lipids that are meant to be absent in control. And this is obviously at this stage, but we want to push it to stages going even further than that. So the hypothesis is there are different lipids that change in Parkinson. They affect the metabolism. They probably affect as Parkinson develops, but there is something very specific about Carnitine Shuttle. So Carnitine Shuttle is our body's mechanism of providing us energy. Whenever your heart, for example, you go for a run and your heart needs lots of blood, lots of energy, Carnitine Shuttle makes sure that all the fats are broken down and you get your sugars and you get your energy so that you keep doing what you're doing to make your heart pump so fast. What happens in terms of Parkinson is this particular shuttle goes faulty, which means it can't change your lipids to fatty acids, and it can't make your fatty acids into sugars that you really need. And when that happens, you get a buildup of lipids. And this theory really nicely, kind of the hypothesis nicely falls with other studies people have done in the past, where they have explained that in Parkinson, the presence of certain kind of lipids is very likely linked to presence of alpha-synuclein. So you might have heard this term before, where alpha-synuclein, a protein, aggregates in Parkinson, and that's thought to be the very first mechanism that causes Parkinson. Now, alpha-synuclein resides in blood, most of it, and there is a very good link between what happens in the blood or what is being excreted in the blood being transferred onto your skin. So things that your blood don't need normally get urinated out, you get rid of it. But sometimes if it's too much in your blood, there are theories which suggest that your blood will be trying to push it out from wherever it can. And one of the pushing out mechanism is through sweat and it's through your sebum production. And this is where what we have done in our research kind of fits in, because if we can detect molecules that are changing because of Parkinson quite early on, then we can detect them on the skin, which is quite far from brain, but it is a very non-invasive way to look at it. And it can be very predictive of something that will happen years after this change is going to occur on skin. So that kind of summarizes what Joy told us. She only planted a bit of seed asking us a question. We decoded the smell of Parkinson using the data first. Then we use multiple approaches to develop it into a test that can be rapidly used to detect Parkinson's. But what has now become out of it is sort of a tree where we are using sebum as a diagnostic biofluid. And particularly in the area of research where I'm involved in tuberculosis, I'm using it to detect tuberculosis before it even happens, or before we get a microbial test for tuberculosis. So briefly, I'll talk about how I've used it in tuberculosis before I end my talk. This concept is same using sebum, but this time what we did was we asked Joy to smell samples from people suffering from tuberculosis. And she could smell it, not a surprise to me anymore, but she could smell the difference in tuberculosis. So what we did was I took Joy to our collaborators in Tanzania, in Africa, and she sat in a TB waiting area, and she said the smell in that waiting area is very strong yeast sort of smell. It smells of like, when there is freshly baked bread and you can smell that yeast. She smelled something similar. And then we gave her some samples from tuberculosis. So when she smelled tuberculosis negative sample, she said it just smells like ordinary breath sample, like a really moist sample. Then we gave her a sample with a really low amount of tuberculosis bacteria, and she said it smelled very salty. Then we gave her TB grade 1 and 2. So these are the ones you can do microbial culture. In about 3 to 7 days, you'll get a test back saying that grade 1 or 2. And she said it was salty, but it was like saline water. And then we gave her a really bad TB. So this is when someone comes to a clinic and they'll be coughing the lungs out and you know something's wrong with them, that kind of TB. And she said it smelled like pus, like leg ulcer, when you get an ulcer in your mouth or your leg, and you know the kind of smell it gets. So a really bad smell. And based on that data, we thought it was quite interesting. So she was able to kind of differentiate if someone didn't have TB with 100% accuracy. Every time someone didn't have TB, she knew straight away. But she also knew if someone had really bad case of TB. Now, that's not really interesting because if someone's got a bad case of TB, they'll be coughing the lungs out and it's easy to spot them. If someone has no TB, if someone's healthy, you don't really need to check them. So we really wanted to focus on the in-between grades when you can't get a microbial result. So putting it into perspective, she was still okay in terms of grading TB grade 1 and 2, where she was only confused if it was grade 1 or 2. So she can still detect if TB was 1 or TB was 2, but sometimes she got confused between 2 and 1, which is fine for a first test. But what we are doing now is we are taking that further. I'm working with Apopo. It's a non-profit charity and organization that works with rats. And they use these rats to smell landmines and TNT and bombs. So the rat go around on lands and they can sniff and they can say if there is a bomb underneath. And then obviously you get bomb disposal to get rid of bomb if it's not already exploited. But now there are no bombs in Tanzania. So they're using these rats to smell tuberculosis. Obviously they smell the presence of Mycobacterium tuberculosis on human. And then they do a specific behavior to say that these are people who have TB. So to end the goal for a global pandemic, a global epidemic of TB by 2035, this research is quite important because right now, if children get TB, there is no way to find out because they won't be able to produce sputum, which is quite important to kind of diagnose TB. So you either have to do a blood test, which will be a week. And if they're going to start spreading TB, it's never going to stop. So with Joy and children and rats, the idea is to use the same platform we discussed with Parkinson to first detect the smell of tuberculosis in molecules, and then try and create something new and smarter, which is a wearable patch. So I'm working on creating a wearable patch that can be put on skin that will detect these metabolites in real time as they are expressed in sebum. So anyone in Africa could put on this patch on the skin, and in 24 hours, the patch will give us the information based on the online processing and online machine learning, the chemometrics part, and tell us if their sebum is something that indicates that there is an infection in the lung or there isn't. So again, this might sound far-fetched, but the idea is novel, but not the application. Currently, people already do this to try and detect cancer. So if there is a mole on your skin, if it's very defined, it's only a benign mole, but if it starts getting edgy, if it spreads, it's cancerous. And people use the same technology on skin to try and identify this sort of mole. But obviously you can see the difference. That mole is something that's physical, your eyes can see it. Whereas what we are going to look into sebum now is not something you can see, but something you can measure. So this will change over time, but it will indicate if someone's got TB or not. And obviously, none of the talks that we do these days are complete if we don't talk about COVID-19. So the same approach with sebum, we also used in one of our COVID-MS coalition. So this is a coalition of about 500 scientists across 25 different countries. And we all had a common goal when COVID outbreak started, was to try and find ways in which you can use mass spectrometry, the techniques we all knew how to use, to detect and standardize ways to find infection of COVID outbreak. So in one of the similar studies that we did with our collaborating university, we also found markers of lipids on skin surface using sebum that are more predictive, and they change more than something like C-reactive protein that's routinely measured to detect the infection. So whenever you got any infection, not just COVID, people look at CRP. But you can see lymphocytes and triglycerides that we detected on our, the triglycerides, especially that detected on the skin, have a much higher response in terms of detection of COVID. And using this information, what you can do is you can tell if someone has got COVID or if they don't, just based on a very simple skin swab that collects the sebum sample. So this kind of comes back to the tree that I told you about a few slides back, that the simple idea that Joy had in 2017 is now a massive tree where we are using sebum to not just look at Parkinson or smell, but something that indicates what's going on in terms of diseases. And hopefully, like I say, this will end my quest for StraTech tricorder, and we might be able to make devices that are like tricorder that people can shine at their skin and they can detect diseases like Parkinson, tuberculosis, and this could probably be available on things like eBay. So make health diagnostics really cheap, accessible, and fast is possible if what we are doing keeps going the way it's going. And with that, I would like to thank you all for being so patient and being still here and not leaving halfway through the talk, and also Stimulus for asking me to come and talk to you. But more importantly, all the collaborators I've worked with so far. So on top, you see our team Parkinson, Perdita Baran, who was the PI on the project. Tilo and Monty are the two clinicians who helped us throughout making sense of our chemical data to put in perspective of biology. Dipanjan to help us develop the paper test that I mentioned earlier. And obviously, Joe and Les for starting the whole project with Parkinson's. Our funders so far have been Manchester University Innovation Factory, Michael J. Fox Foundation, and Parkinson's UK for Parkinson's work. I'm working with Steve Fowler, who is an expert in respiratory diseases. And my study with APOPO is funded by MRC, the Medical Research Council in UK. And I worked with Mel Bailey at Surrey for the results I showed you for our COVID work. And finally, while you think of the questions you want to ask me, I'm going to leave you with a thought about science, which I truly believe that no good science has ever come if one person was going to work on his own trying to discover. Like you've seen in most of my research, it's been a joint collaborative project. But beyond just scientists, I think most of the good discoveries in health diagnostics are going to not just come by scientists working together, but it's going to come by end users, people like yourselves who have novel ideas, and everyone, not just analytical scientists, medical scientists, but everyone who can put the brain together to get new ideas and people who can execute it is going to bring out new science and new discoveries that we are after for health. That's it. I'm going to stop talking now, and I'll let you guys ask me questions if you have any. I'm going to stop the share.
Yeah, thank you so much for that amazing talk, Dr. Trivedi. I'm Stuti, and I'm one of the co-founders of Stimulus. And thank you so much for joining us today evening. I believe it's quite early evening for you over there. So I had a small question, and before I thank you, I would like to ask the question. So I'm a clinician. I'm working as an occupational therapist, and I see patients with Parkinson's disease quite often on day-to-day basis. And so and they're probably usually towards the more severe end of the spectrum, where they're more progressing towards stage 4 to 5. And so, like you correctly mentioned, it's quite a challenge to live with Parkinson's disease. So I was wondering if if you've given thought into, or whether it might be possible to expand the diagnostics to other neurodegenerative conditions as well, like Huntington's or Alzheimer's or frontotemporal dementia or things like that?
I think it's very much possible. The reason being, like Parkinson's, things like Alzheimer's and Huntington's, they are all the primary thought of why they occur and what's causing them is development of proteins that either fold badly or they develop badly. So if the proteins are the cause of these diseases, and if you found a way to look at the effect of this protein change quite early on before they go bad on skin surfaces, then I don't see why we cannot adapt the same pathway or the same kind of approach to look at the effect of a different protein on skin. So it's very much likely.
Right, that's very interesting. Thank you. Do we have any other questions from the audience?
Yes, I do have question. Can I ask it out loud?
Yes, please do.
Hello, thank you so much for such an amazing lecture. I have a question about the potential treatment of Parkinson's disease. Right now, the amazing tool CRISPR was discovered in the biochemistry field, and right now it is successfully implemented to treat several inherited diseases in animals. And I just wanted to ask you whether it is possible to implement the same tool to treat human inherited diseases, such as Alzheimer and Parkinson.
A difficult question. The reason being, CRISPR is, it's developed quite rapidly than what it was predicted to, in the pace at which it should have developed, or people thought it would. So CRISPR is doing some amazing tasks in terms of editing gene and making sure that we have good results that we rely on. But how much it can be translated, and not just translated, but also be helpful for treating Parkinson's, it's a very much gray area. The reason being, if you do modify a gene, and if you can, if you can tackle alpha-synuclein, and if that's the only thing that's gone wrong in Parkinson, you have a cure. But if you can't, and if there is more than one interaction, so if it's not just alpha-synuclein, and one of the train of thoughts in Parkinson is alpha-synuclein is not the cause, but it's the effect of something else going wrong. Because the buildup of Lewy bodies, it's quite a researched area, and people have different thoughts the more they study it. So one of the ideas is if it's the effect, then all the tools we are right now putting towards the cure by tackling alpha-synuclein and Lewy bodies might just be tackling the effect. So it's like treating the symptom, but not the actual cause of this symptom. But based on what I've studied, we are quite far from cure ourselves, and I don't think we probably have cure, but we might have more understanding of what's going wrong rather than if you can cure it. So someone else might have to come up with the cure, but probably with the research we are doing, I think we'll be able to tell more about what's going wrong other than alpha-synuclein.
So, but can I just ask another question about this topic? You said that it is like it would be very complex for us to treat Parkinson's disease with a CRISPR tool because we will be treating symptoms, not like the cause of the disease, right? But would it be possible to probably treat fetus during the woman pregnancy stage and eliminate or treat the disease gene in the genomics of this like little child?
I think very possible, but I don't know what the ethical complications we might end up with, and it might never be accepted ethically because it's maternal decision for the child. So it's the same thing that I faced when I was looking at Down syndrome. So this was one of my Viva questions in my very second year of my PhD. And I was asked that all your research is fine, all you're doing is fine, but the implication that you're indicating is you'll be able to detect if the baby is going to have all normal chromosomes or an extra chromosome, which means that you could either treat it or it's too late to treat it and the mother could decide to abort a child. So ethically, how do you think your research is? And it's the same question. Most of your science depends on ethics. So is your science going to be accepted ethically? don't know.
Mhm.
But in terms of, is the science possible? Yes.
Okay, thank you so much. Would it be possible to email you with any further questions? Yes, thank you.
Yeah, sure. On my slides, I had my Twitter and email both, so whichever you use, it's fine.
Okay, thank you.
Thank you, Elena, so much. And I'll be sharing Dr. Trivedi's contact details in the Discord channel, if that's all right with him.
Yeah, that's fine.
Okay, so I'll be putting his email address in the Discord channel so that if anybody else has any other questions, you can email him. Or does anybody else have any other questions? Right. So I guess Stuti.
Yes, thank you so much, Dr. Trivedi, for taking time and talking to us. I think this is the first time I've attended a talk which has looked at for Parkinson specifically, like in terms of diagnostic biomarkers. I've mostly attended a large majority of talks which have looked at neuroimaging biomarkers. So this is quite novel, and I really learned a lot, and I'm sure that all our attendees did as well. On behalf of Stimulus, and all the members of Synapse Society, I would like to extend a very heartfelt thank you to you. And I think this is I think you're one of the first external speakers for our Synapse Society. And as Arya introduced to you in the beginning, we started the initiative of Synapse Society with the intention to expand knowledge about the neuroscience and break down the barriers to neuroscience and the brain sciences, and make it accessible to everyone. So I think many of the students who attended today are as young as class 12 students as well. So I think it was a great opportunity to listen to you talk and share your science with us. And I completely agree with your last slide that science is all about collaboration. And hopefully with talks like this and with Synapse Society, we can get those conversations started as young as possible and as early as possible. So thank you for being a part of making this possible. And yeah, thank you so much for coming.
Thanks for inviting me. It was a pleasure, and I think it's a great initiative, and I hope it goes really well as you're planning, and you bring out really good collaborative scientists in 5 or 10 years.
Thank you so much. And to all the attendees also, thank you for attending, and please keep the Discord server active and keep checking it, and we will be posting information regarding further talks and events on the Discord channel.
Thank you so much, Dr. Trivedi. I'll be ending the meeting now, but if anybody else has any questions, okay, they can put it in the Discord channel or you can email Dr. Trivedi. Thank you so much for joining today's lecture. Have an amazing weekend ahead. You too, Dr. Trivedi. Thank you so much. Have a good evening.